Okay. Okay, let's get started. Good afternoon, ladies and gentlemen. My name is Chavirakarn Manphibool from Chulalongkorn University, Thailand. Today, I'm going to present our research, the effect of light-emitting diode toothbrush on Streptococcus mutans biofilm in vitro. Let's get started with the introduction. Streptococcus mutans or S. mutans is a common type of microorganisms found in dental plaque. It is also known as the potential pathogens for demineralization that cause dental caries because they metabolize dietary sugar to acids. According to the ecological plaque hypothesis, this microorganism normally exists in a small amount even within a healthy host. However, when the oral environment critically changes, such as high fermentable sugar or low salivary flow, the pathogen will outgrow and cause dental caries. Nowadays, many studies show the connection between blue light and antimicrobial activity. Blue light is the visible light, which its wavelength is about 400 to 500 nanometers. The investigations.
Excuse me.
Sorry.
Excuse me.
Yes.
We cannot see your slide. Could you please share again, please? Sorry.
Really?
I can see your face, but I cannot see your slide.
From the start? Can you see it?
Yes, yes.
Okay, from the beginning, right?
Share your slide again. Yes, now you can see. Okay.
I'm sorry.
Yes, yes. Thank you. Sorry that I noticed you too late. Okay.
Okay. Let's get started. Good morning, ladies and gentlemen. My name is Chavirakarn Manphibool from Chulalongkorn University, Thailand. Today, I'm going to present our research, the effect of light-emitting diode toothbrush on Streptococcus mutans biofilm in vitro. Let's get started with the introduction. Streptococcus mutans or S. mutans is a common type of microorganisms found in dental plaque. It is also known as the potential pathogens for demineralization that cause dental caries because they metabolize dietary sugar to acids. According to the ecological plaque hypothesis, these microorganisms normally exist in a small amount, even within a healthy host. However, when the oral environment critically changes, such as high fermentable sugar or low salivary flow, the pathogen will outgrow and cause dental caries. Nowadays, many studies show the connection between blue light and antimicrobial activity. Blue light is the visible light which its wavelength is about 400 to 500 nanometers. The investigations about the bactericidal effect of blue light on S. mutans and oral biofilms suggested that it could reduce the S. mutans viability and inhibit biofilm development. The LED blue light is recently incorporated into electric toothbrushes for various purposes, such as to improve of oral hygiene or tooth whitening. So, what is an LED? LED stands for light-emitting diode. It is the process that the light is released after the current is flown through an inorganic material called semiconductor. The specific type of semiconductor that mostly used to generate blue light is gallium nitride. However, there is no study yet that examines the effect of an LED light sourced from the LED toothbrush to the oral biofilm. So the objective of our study are 1. To examine the bactericidal effect of LED toothbrush on Streptococcus mutans biofilm, and to compare the bactericidal effect of different exposure time of LED toothbrush on S. mutans biofilm in vitro. Next is our material and methods. The LED light source in our study was from the LED toothbrush. The trade name is White & Girl UK, which was certified by European Conformity or CE marking. The specifications indicates 460 to 480 nanometers wavelength, which is the blue light, as I mentioned, and 840 milliwatts of power. We also measure its radiance. The LED light located at the toothbrush head, covered by the silicone bristles, which were 9 millimeters long. For the bacterial growth, we use S. mutans UA159 strain from bacterial gristalostops were incubated in brain heart infusion agar or BHI agar, and then were incubated at 37 degrees Celsius with 5% carbon dioxide for 24 hours. The isolated colony was regrown overnight in BHI broth with sustained shaking at 240 rounds per minute. After that, its optical density at 600 nanometers was evaluated and adjusted to 0.1. The culture was then incubated in the proper condition for 3 hours to reach the determined logarithm phase of growth, or OD 600 nanometers, around 0.4 to 0.6, which would be used for the biofilm formation. For the biofilm formations, the cells were then resuspended in BHI broth with 1% sucrose. The 3 milliliters of suspension, which contains 3 times 10 to the 8 bacterial cells, were added into each culture plate and were incubated at 37 degrees Celsius with 5% CO2 for 24 hours. The biofilms were then divided into 5 groups: the control group with no LED exposure, and the experimental groups exposed to the LED light for 15, 30, 60, and 120 seconds. Each group contained 2 plates of S. mutans biofilms. The biofilms were then scraped off and put into 1 milliliter of phosphate buffer saline or PBS. The bacteria in PBS suspensions were sonicated and serially diluted from 10 to the -1 to 10 to the -8. An aliquot of 100 microliters of its concentration was then sprayed on BHI agar. The concentration that yields 30 to 300 colonies will be used to count the number of bacteria. The experiments were repeated 3 times with duplicate sample per group in each experiment, so the total N in our study is equal 6 per group. Here is our results. The percentage of survival was calculated relative to the control. The cross-cover lid test was performed to compare the bactericidal effect of blue light from the LED toothbrush among groups. Statistically significant difference was set at p-value less than 0.05. Post-hoc comparisons were carried out by Mann-Whitney U-test with Bonferroni correction of the significance level of multiple pairwise comparisons, or p-value less than 0.005. Okay, this graph depicts the percentage of bacterial viability. Our results show that exposure of LED for 120 seconds significantly decreased the percentage of bacterial viability compared with those exposed for 15, 30, and 60 seconds and a control group at p-value less than 0.005. Next is our discussion. The mechanism of bactericidal effect of light is based on the photosensitizer, or PS in this picture. It is the agent that can absorb the light. When the photosensitizer is activated by light with its preferable wavelength, the electrons are transferred to produce the radical ions that react with the oxygen, result in cytotoxic species. As I have mentioned, that previous study revealed that blue light could reduce S. mutans by biofilm reformation. Our data supported that the visible blue light from toothbrush could reduce viability of S. mutans in biofilm. And the blue light in LED toothbrushes in our study had to pass through the silicone bristles, So it is important to test whether the LED light in the toothbrush could still have an antibacterial effect. Our results suggest that the light from the LED toothbrushes can significantly reduce S mutans viability in biofilm after a 2-minute exposure. Another point is that the exposure time of blue light on bacteria is one of the most important factors for bactericidal effect of LED toothbrush. Previous in vitro studies set the exposure time from 1 to 10 minutes. However, people normally brush their teeth only around 2 to 3 minutes. Our data show that a significant reduction in bacterial viability occurred after the biofilm had been exposed to LED blue light for 120 seconds. According to the recommendation of American Dental Association, toothbrushing should be performed for 2 minutes, twice a day. So, if an LED toothbrush was applied for 2 minutes, it may help to reduce bacterial viability in the oral cavity. However, the LED toothbrush in our study was applied to the biofilm formed in 35 by 10 millimeters culture plates, which have smaller surface area compared with the oral cavity. In conclusion, the LED blue light from LED toothbrush significantly reduced the number of S mutans in biofilm in vitro when the biofilm was exposed to the light for at least 120 seconds. Thank you for your attention.
Thank you very much, Dr. Chavirakarn. Thank you for your very interesting presentation. Okay, so after hearing your presentation, I would like to buy the LED toothbrush.
Really?
It's also available now, right? You say that the name is White Gold.
It's not available in Thailand yet, but generally provided in Europe and America and even in Korea, I think. Yeah.
Okay.
Yeah.
Do you have any recommend if we use in the mouth? As you said that for toothbrushing, normally they recommend for 2 minutes for brushing. But if we use this type of toothbrush, how many minutes that should we use?
I think
for full mouth brushing. Okay.
For the available data and my research, at least you need to brush your teeth for 2 minutes to reach the effectiveness of the LED toothbrush. But in my opinion, you don't brush your teeth only in the one area for 2 minutes, right? I mean, in the quadrant 1, quadrant 2, 3, and 4, it's not 2 minutes. So I think more data in clinical study should be performed to confirm this issue. But nowadays there are only 2 or 3 clinical studies that test the effectiveness of the LED toothbrush on the gingival index and dental plaque or stuff.
Okay, and for the LED concentration, how to calibrate the LED light concentration?
Actually, I did not calibrate it, but I measure the radiance at the Faculty of Science at Chulalongkorn University, and the power is equal to the other trade name, and the trade name that had the clinical study. The power is equal. Yeah.
Okay. Okay. Thank you very much for your answer.
Okay.
Okay.
Thank you.
Is there any question more?